This is Alejandro, a man living in rural Peru. One day while asleep he receives a bite from a kissing bug which he instinctively rubs with his finger. He thinks nothing of it, the bite heals, and he continues to live his life. 20 years later, he suddenly dies of heart failure. The suddenness of this death is all the more tragic. However, little did anyone know that 20 years ago that bite had become infected with the parasite Trypanosoma cruzi, which is responsible for the Chagas disease, which ultimately led to his heart failure. Was this preventable? Could the disease have been identified? What treatments are available for the Chagas disease? All this and more will be answered in this video covering the Chagas disease, including a small molecule case study on BENZNIDAZOLE, one of the only commercially available drugs with efficacy for treating the Chagas disease. The Chagas disease is endemic to Latin America and classified as a neglected tropical disease. According to the World Health Organization, the Chagas disease is a proxy for poverty and disadvantage, affecting populations with low visibility and little political voice. The Chagas disease affects a predicted 8 million people, most of whom do not know they are infected. Progress has been made to interrupt transmission vectors. However, in many places, vectorial transmission has not been interrupted, shown in red. The Chagas disease is caused by a single-celled parasite called Trypanosoma cruzi, or T. cruzi for short. This is a flagellar eukaryotic organism, meaning it possesses a flagellum and discrete organelles such as mitochondria and a nucleus. T. cruzi feeds on nutrients in the host by diffusion. The main transition vector of the Chagas disease is the Triatomini subfamily of insects. These feed on vertebrate blood using a piercing beak. These insects often feed on nesting animals and livestock. A few of the Triatomini species are adapted to live in human environments and are the main cause of concern for transmission of the T. cruzi parasite in the human population, causing infestation around people's homes. Bites often occur near one's mouth or eyes, giving the common name kissing bug. A kissing bug becomes infected with the T. cruzi parasite when it drinks the blood of an infected animal or person, ingesting the parasite. Inside the gut of the insect, the parasite differentiates into an epimastigote, which can replicate by binary fission. The parasite then differentiates into the trypomastigote form capable of infecting a new host. As the bug feeds, it defecates, leaving the parasite near the fresh wound. The parasite enters the body through the bite or through the eyes if the area is rubbed by the victim. Once inside the body, the trypomastigote penetrates cells, then differentiates into amastigotes, which replicate by binary fission. After replication, the parasite differentiates back to the trypomastigote form, bursting out of the host cell. The trypomastigote is responsible for spreading the infection to adjacent uninfected cells, or to distant sites in the body through the bloodstream, consolidating the infection. Subsequent bites received by an infected host cause the spread of the disease to other people, and the cycle continues. Other less significant transmission vectors include donation of infected blood or organs and ingestion of the parasite through contaminated food. Prevention of infection requires control of vectorial transmission and screening of blood and organs for donation. Eliminating Triatomini, the primary transition vector, has been achieved in some areas of Latin America by use of insecticide spray and with home improvements such as placing netting on windows and vents. This can be effective at interrupting the spread of Chagas disease by the main vector. T. cruzi is a parasite, therefore it is adapted to evade the immune system and cause minimal damage to the host. If the host lives longer, the parasite has a better chance of proliferating. This is in contrast with viral and bacterial diseases which multiply much faster and can be transmitted by direct contact, giving them a much shorter life cycle and less dependence on the lifetime of the host. T. cruzi evades an immune response during infection, 1. by invading host cells, and 2. by having a highly diverse surface antigen expression causing a cloud response of many different antibodies, and polyclonal B cell production, delaying the production of an effective and specific antibody, and 3. by possessing defenses against toxicity, with antioxidants to eliminate toxic species produced during phagocytosis or that are produced by neutrophils. Due to this slow immune response, The initial introduction of the parasite leads to infection of cells, and the multiplication of the parasite within the cells, which leads to many more parasites being released after the first 24 hours of infection. The Chagas disease has two phases. The first is the acute phase, characterized by parasitemia, allowing the parasite to spread throughout the body while the immune system struggles to suppress the infection. The acute phase lasts for 4 to 8 weeks before the immune system can suppress the prevalence of the parasite in the blood. Enter the chronic phase. Although the parasite has been eliminated from the blood, it has already proliferated and infected tissues around the body, where it will remain indefinitely. For white blood cells to mediate an immune response within tissues, inflammation occurs where the blood containing white blood cells is ejected into the tissue to fight the disease. Unsuccessful attacks and persistence of the parasite leads to long-term inflammation of the infected areas. This long-term inflammation causes tissue damage and the buildup of scar tissue. T. cruzi preferentially infects the heart, where the long-term inflammation damage leads to cardiomyopathy, the stiffening of the heart walls, reducing the ability of the heart to pump blood. Treatment of the Chagas disease requires diagnosis. During the acute phase, diagnosis can be achieved using PCR and microscopy of the blood samples. Quick diagnosis during the early stage of the disease allows for more effective treatment. PCR is the process of amplifying and testing for a specific type of DNA. In this case, the DNA of T. cruzi is of interest. Later in the disease, diagnosis and treatment is more difficult. The parasite becomes eliminated from the blood, therefore PCR and blood microscopy have low sensitivity. Antibody and antigen tests such as ELISA are effective at determining the presence of the Chagas disease. BENZNIDAZOLE is a drug registered in 1971 with the application of eliminating T. cruzi infections. Treatment efficacy with BENZNIDAZOLE varies depending on the age and condition of the patient. Treatment courses for the acute phase typically last 30 to 60 days, with 2 oral doses a day due to the 12-hour half-life of the drug, totaling 200 to 300 milligrams a day. For acute and early infections, the cure index is quoted to be around 60% with BENZNIDAZOLE. Treating later stage and chronic infections has a lower success rate. Various small studies conducted between 1980 and 2000 show little significant difference between groups treated with BENZNIDAZOLE and the control. For example, one study using ECGs or electrocardiograms to monitor heart palpitations found heart problems in 5.3% of treated patients, versus 22.8% in untreated patients. However, the low number of heart issues presented in the study, 20 out of 200 test subjects, and the similar prevalence of T. cruzi in both the treated and the control make interpretation of these results inconclusive. In general, it is found that the parasite can persist within organ tissue even after treatment during the chronic phase. BENZNIDAZOLE treatments are discontinued due to unpleasant side effects in about 20% of cases. Symptoms range from nausea and vomiting to damage of periphery nerves, usually occurring after 12 days of treatment, in which case the treatment should be suspended. One study showed that a 2-week course of BENZNIDAZOLE to treat chronic phase infections could mitigate the severity of side effects by being so short. This was also effective at eliminating T. cruzi from the blood. However, there is a bias in this study caused by using only PCR-positive individuals, since most chronic cases do not yield positive PCR tests nor parasitemia. Positive PCR suggests a persistent parasitemia, possibly caused by a weak immune response to the parasite. The resulting reduction in PCR intensity suggests an elimination of the parasite from the blood, shown on the graph, but not particularly from tissues. Treatment during the acute phase is more effective because the parasite has had less time to consolidate the infection. The drug assists the immune system in eliminating the parasite from the blood more quickly and helps kill minor tissue infections. The IC50, or concentration at which half of the T. cruzi population dies, is 3 micrograms per milliliter. A pharmacokinetic study showed that the peak blood concentration of BENZNIDAZOLE is 2.2 micrograms per milliliter after a 100 milligram dose, suggesting that the drug is effective at killing blood-bound parasites. However, as diffusion distances in the body increase, so does the volume occupied by the drug, reducing the BENZNIDAZOLE concentration. Therefore, treating the chronic phase is less effective because the parasites have manifested deep in tissues. How does benznidazole work? The nitro group on the benznidazole binds to and is reduced by nitroreductases within T. cruzi. The aromatic species then undergoes a series of reactions, producing a glyoxyl species, a dialdehyde that exhibits a high reactivity and can oxidize important biomolecules within the cell. A study suggested that glyoxyl oxidizes free nucleotides within the cytoplasm of T. cruzi, becoming incorporated into the genome and causing double-strand breaks. Although T. cruzi has DNA repair mechanisms, too much damage to the DNA causes cell death. Another study showed the high affinity of benznidazole for binding with NADH fumarate reductase, acting as a competitive inhibitor, preventing the production of an important biomolecule in energy production in T. cruzi. The factors contributing to the toxicity of benznidazole on T. cruzi are not fully understood. The reduction of the nitro group is well characterized, but the production of glyoxyl is slow, suggesting that there are other pathways to producing reactive species that could interrupt RNA and protein synthesis. Benznidazole is also metabolized in human cells, also producing reactive species. This accounts for the low half-life of the drug and the adverse symptoms experienced by patients. This metabolism probably prevents the drug from reaching the target parasite within tissues, accounting for the limited treatability of the chronic phase of the Chagas disease. Prevention is the most effective way to mitigate proliferation of the disease. However, for those who are infected, the current available treatments leave a lot to be desired. As previously discussed, a vast majority of people who are infected with T. cruzi are not diagnosed. As screening for the Chagas disease improves, the number of people that need treatment in later stages of the disease will increase. Further, vaccination would be very difficult to achieve due to the high variety in the cell surface antigens exhibited by T. cruzi. Improving the specificity of the drug could improve the effectiveness by avoiding breakdown of the drug in human cells and having a higher toxicity to the parasite. One way would be to design a highly selective drug that inhibits a specific metabolic protein only found in T. cruzi. The biggest problem with the Chagas disease is the increase in premature deaths due to heart failure. Eliminating the parasite from the heart after a chronic phase diagnosis would prevent the long-term inflammation and build-up of scar tissue, causing myopathy. Instead of targeting the parasite, an antibody-based drug delivery system could be employed to target the heart. By connecting a drug such as benznidazole to a specific antibody, you could target the heart, whereby the antibody is injected into the host's bloodstream, accumulating in the heart by binding to organ-specific cell surface antigens. This would allow for higher concentrations of drug at the specific site, eliminating more of the parasite than if the drug were ingested. This could prevent the side effects that occur due to oral ingestion of benznidazole, making the treatment more tolerable. The higher drug concentrations will allow for shorter treatment courses. The downsides of this treatment are that the drug must be injected into the bloodstream, as antibodies are broken down by the digestive system, meaning that administration at home would not be viable. Side effects could occur due to the immune system attacking the introduced antibodies. This is a relatively new area of medicine, so an effective antibody-based drug for treating Chagas disease could be much further in the future than a more traditional simple molecule approach. And finally, this approach would not eliminate the T. cruzi infection from the whole body, just the heart, meaning reinfection of the heart would be a possibility.